Welcome to our video for week 1 of our course. We are going to, in this video, review our content for the week along with our weekly expectations. So if you remember in Brightspace, under the banner for the week, all of your expectations are listed here. This is going to be your week 1 content video. The content that I'm going to be reviewing is from this PowerPoint, which is linked here. That's titled week 1 and then our course information. So we're going to go into that PowerPoint now, and then we'll touch back on some of these other requirements for the week. So, week 1 covers 2 chapters. We are going to be learning about the genetics, underlying developmental disabilities, and fetal development. As you look through and read through these PowerPoints, you need to be on the lookout for our university seal. The seal is going to be the clue that you need when you are working on your reading quiz. Okay? These are those objectives again from the week that are on Brightspace listed here. Now, starting with this content, I wanted to tell you that our textbook is very, very intense and in-depth about a lot of scientific topics. Our textbook goes very granular on those things. I do not want you to overwhelm yourself with trying to get every piece of information out of each chapter. I just want you to kind of read your chapters thinking, what information will I need to help me become the best educator possible for my students? And sometimes that's not necessarily knowing really, really specific things about the cells in our body. So I want you to keep that in mind. Try not to stress or freak yourself out about this content. If it's important information, I will let you know that it's important. If it has the university seal on it, pay close attention to it. But other than that, just take it at face value, okay? So chapter 1 kind of gets into all about like what makes up humans, what makes up us. And it talks about how we have our human karyotype is made up of 23 pairs of chromosomes. The male chromosome breakdown is here and the female chromosome breakdown is here. And each of these pairs makes 46 individual chromosomes. And our human cell looks like this, and it has all of these parts and pieces to it, and if you see there, each cell has DNA in it, and that's kind of the code that makes us be expressed as who we are, what you can see on the outside, and also what is happening on the inside of our bodies. Our chapter goes really in depth about mitosis and meiosis, and these are how our cells split and make new cells, okay? So mitosis is when cells are regenerated. This is when a cell duplicates its DNA and divides into identical copies, okay? Meiosis is a specialized cell division that reduces the number of chromosomes by splitting them in half. This is when sperm and egg cells are produced. When there is meiotic non-disjunction, this is when people are born with too many or too few chromosomes. And we're going to talk about those disorders as the chapter goes on. As a fetus is developing, their cells are duplicating, regenerating, super fast. Extremely fast. And if there is an abnormality, a chromosome at the chromosomal level, a majority of these fetuses are spontaneously aborted. So that is something that happens frequently when there's something off about your cells duplicating and regenerating. This is another example of how mitosis and meiosis works. When we have a cut on our finger and our cut is healing, our skin cells are going through mitosis. They are duplicating themselves, and it's the exact same cell, right? It's the exact same DNA copy. But when our body is producing sperm or an egg cell, it's going through meiosis, that type of cell division. In general, in our general population, a chromosomal error causes disorders that occur in 6 to 9 per 1,000 of live births. So not a large amount, but these chromosomal errors result in a lot of the disabilities that you will be encountering in your career. As women age, the risk of errors in meiosis increases. That's why a lot of the medical recommendations are that women do not wait too terribly long to have children, because as they get older, errors in this cell division can cause errors in their eggs and errors in their babies potentially. Almost all of the growth that occurs in a fetus is occurs through mitosis, which is that cell division that's creating your bones, your organs, all of the parts of your body. If you are curious about learning more about mitosis and meiosis, this is a very helpful video. You are free to watch that on your own. Now, we'll jump into chromosomal abnormalities. This is an example of all of the ones that most of them are touched on in this chapter. So when our cells are dividing, these abnormalities just happen, okay? And one of the ones that happens is you might gain a chromosome. This is what is known as trisomy 21, an example of this. That's when you might have 3 copies of chromosome 21, and that is also known as Down syndrome. You might lose a chromosome during this division, and that is known as, an example of that is known as Turner syndrome, which is females with only one X chromosome. Mosaicism is when cells have different genetic makeups. They might lose an extra chromosome. 5 to 10% of all of your chromosomal abnormalities are mosaicism. Translocation is when a portion of one chromosome is transferred to a completely different one. So in this picture is a great example of this part of this chromosome was transferred over here, and they kind of did a little swap. This one, they did a swap, but then this part didn't return. It just replaced it. Additional ones are deletions. There are chromosomal deletions, whether it's a part or a whole pair. There's visible ones. That means it's visible under a microscope, so they can look at whatever it may be and see that deletion. An example of that is Cry-Kuchat syndrome. Microdeletions are only detected at the molecular level. And an example of this is shown in this picture of Smith-Magen's syndrome. 25% of eggs and between 3 to 4% of sperm have an extra or missing chromosome. And an additional 1 to 5% have a structural abnormality. So, data tells us that between 10 to 15% of all conceptions have a chromosomal abnormality, and 95% of those do not survive to term. So it's far more likely that a pregnancy that has a chromosomal abnormality will not survive to birth. Our genes is a specific segment of DNA that codes for a specific protein or RNA. Our DNA is that code that tells us, that expresses our genes in specific ways. So abnormalities can also come from these specific genes. Our DNA is made up of 4 different chemicals that are listed here. If you've ever taken a chemistry class, I'm sure you're familiar with that. And the same way that we have chromosomal abnormalities, they also can happen in our genes through transcription. It gets written into the code wrong through a translation. They swap places. Through a mutation, there's might be an insertion or deletion of different genes or different code information. This slide is on Mendelian disorders, which is also in this table in your book, table 1.3. It's on page 13. This review talks about 3 different ways that disorders can show up in our bodies. An autosomal recessive is when there is an abnormal copy on both chromosomes that come from both parents. So this is when maybe both parents are carriers for something, some type of disorder, and then it's a recessive disorder. So the parents might not know that they're carriers for it, and they come together, and this recessive piece from both parents is expressed in their child. This is where we have a lot of enzyme defects that come into play. It is rare in the general population. It's extremely rare, and 1 in 2,000 to 1 in 200,000 or fewer. So because enzyme deficiencies generally lead to biochemical abnormalities involving either insufficient production of a needed product or the buildup of toxic materials, these lead to developmental delays or possible early death. So a lot of these autosomal recessive disorders can alter the way your body processes specific things like specific types of vitamins and nutrients, and maybe even like cholesterol. So your body can't process it appropriately, and that can lead to other issues that cause developmental disabilities in your brain and your body. An autosomal dominant is a dominant type of disorder where it is probably expressed in one or both of the parents. And this is where physical impairments come from a lot of the times. Some examples of this are Huntington's disease or achondroplasia. And then X-linked disorders are when mutated genes are located on the X chromosome, and this primarily affects males. This is very interesting from our text. It says that 10% of males with an intellectual disability and 10% of females with a learning disability are affected by X-linked conditions. And males are more than twice as likely to have intellectual disabilities than females. Because X-linked disorders affects males more frequently, there's an unusually large number of genes on the X chromosome that are critical for our brain development, our nerve, our cell functioning, our learning, and our memory. And up to 10% of all known genetic errors causing intellectual disabilities are on the X chromosome. So it's possible that you might be working with students who may have an X-linked disorder, as that affects a lot of our intellectual development and our learning and our brain work. This is all that information from that previous slide, just from the chart in our book, basically, and it has examples of each disorder. The chapter also touches very briefly on these topics. I'm not going to go into them too much in depth because we'll have whole chapters dedicated to each topic as we go on, but it basically talks about how epigenetics is how our genes express themselves in ways that aren't due to the written code. It's not necessarily our cellular makeup, it's kind of other things that can impact the expression of our genes. Talks about genetic testing, different types of environmental influences, and some types of therapies. So if a genetic abnormality is detected, there are possible therapies that can be used to help increase the quality of life of an individual with a chromosomal abnormality. But we'll talk a lot more about this as the chapters go on. So in summary, the important things to know is that each human cell contains a full complement of DNA, which is 23 chromosomal pairs. Developmental disabilities may be caused by an unequal division of reproductive cells, a deletion of part of chromosomes, a mutation in a single gene, or a modification of gene expression, how the gene's expressed. Genetic testing can diagnose many genetic disorders, and an early identification can lead to improved outcomes as a result of genetic therapies. So this chart from our textbook shows how fetuses are developed and possible malformations that occur and when they are most likely to occur. Throughout the human development, there are several different periods. The first is the embryonic period. This is when all major organ systems are formed, but they're not fully functional yet. So this period is broken up into different stages. The first is germinal. This is when it's between that time period between fertilization and implantation of the embryo. And at this time, cells are dividing at an extremely rapid rate to form the embryo, which becomes the fetus. Gastrulation is when the embryo starts to organize itself into 3 different layers of cells, and that's when the structures and organs begin to develop. And then neurulation is the stage when the neural tube begins to form. This is what will eventually lead to the development of the central nervous system, the brain, and the spinal cord. Then moving into the fetal period, the neural proliferation period is between 6 to 27 weeks gestation age. GA stands for gestational age, and that's the age of the fetus within the pregnant person. So 6 weeks gestational age means that fetus has been growing for 6 weeks. This is when gray and white matter cells start forming in the brain, and these are very important cells for brain development. The neural migration period then starts, and that's when those neurons start to migrate towards the surface of the brain. So like in this image, so they start to migrate towards the surface between 12 to 20 weeks gestation. And sometimes if there's a disturbance in this period, it can cause what's known as smooth brain. So instead of your brain having all the wrinkles it needs, it has smooth. And some other types of disturbances can be caused from this time period, such as seizures. And there's possible links to autism. The cortical organization and synapse formation. Synapses are the point at which an impulse passes from one neuron to another, resulting in a neural activation and communication. This is extremely important for all of our brains. Even now, that's how our neurons talk to each other, but especially when a fetus is developing, they need that communication between neurons. So when this starts in the fetal period, it's very, very important. And sometimes disturbances can happen that make these synapses not as strong or effective or not at all. And we'll talk about how that can lead to some abnormalities. Myelination, you'll watch a video on this. It is a neuron insulation that facilitates speed and coordination of these transmissions and formations. So it's kind of like this insulation, like how we insulate our house. It insulates the neuron to help this like communication. And so stronger the myelination, the stronger and more successful the communication is. This myelination reaches its peak during the first year of postnatal life, so the first year of the baby's life. But it's starting when the baby is a fetus. The placenta is an organ that is extremely important to the mother and the baby, especially in baby's development. So if you see this image of this baby, and then this is the placenta, this is where the umbilical cord is linked to the baby. The umbilical cord is linked to the placenta, so the mother's blood gives oxygen and nutrients to the placenta. The placenta gives that oxygen and nutrients to the baby, and then any waste that the baby has, that carbon dioxide, goes back through the placenta. So it's this passive transfer of nutrients from mother to baby. So it's very important when a mother is pregnant that she has good nutrition, and she is conscious of the things that she's putting into her body, because that can affect the development of a fetus. Something else that's passed is immune cells. The placenta activates a maternal immune system, so it helps keep the mother healthy, but it also is protecting the baby. So because the fetus is immunocompromised, that's why the placenta helps the mother stay nice and healthy. But if the mother does get sick, passive immunity can pass to the fetus. That's why a lot of times specific immunizations, specific shots are recommended during a pregnancy, because those antibodies can also get passed to the fetus during pregnancies. So the environment of the baby in the inside of a mother greatly influences their human development. So this starts even before a mother is pregnant. So this is called the preconception period. Things that are impactful are parents' age, both sets of parents, their health, their nutrition, and their environmental exposures. If they're exposed to environmental toxins that are not optimal for a successful fetus, then that's going to affect the fetus as it continues to grow. The embryonic period, when that's an embryo before it becomes a fetus, it's that kind of ball of cells. There are any deviations from normal development during this period can result in major structural malformations. So issues with the structure of a fetus, whether that's organs, bone development. Up to 80% of all miscarriages occur during this period, the embryonic period. And there's several genetic mutations that can occur in the brain during this period as well. During the fetal period, they are vulnerable to disruptions in the typical growth and developmental of existing body organs. And there's a lot of brain work that can be altered during this time period that can lead to developmental disabilities. So it's very important that the mother keeps herself healthy, keeps herself with good nutrition, keeps herself in a good health, and decreases her environmental exposures. The fetus can have an injury that can result in a disability that you may encounter. Fetal injury, some of them that occur, one of them being an infection. Congenital cytomegalovirus is the most common fetal injury. It's listed, it's kind of this nice infographic right here, gives a little information, but it's an infection that can cause birth defects. So issues with baby's skin, baby's liver, how they're processing their blood and their urine, and maybe even their physical shape of their body, and also abnormalities that might last further past birth. There can be inflammation among the baby. There can be issues with their heart. There can be issues that result from substance abuse from the mother, including alcohol, nicotine, smoking, cocaine, stimulants. We will have a whole chapter on environmental exposures. And also, mother stress and anxiety and depression can lead to injury on the fetus. That's why prenatal care is so important. Prenatal care is the care that mothers get when they are pregnant. So it's very important that a medical professional evaluates fetal development. So they get medical history, including genetic and environmental factors. Mothers get screening, they get ultrasounds, blood work, genetic testing. They are constantly watched during this time period because if there is an abnormality or a risk factor or a genetic issue that is caught earlier or identified earlier, there can be different pathways that can be taken to support the mother and support the fetus. Some diagnostic considerations. Is prenatal testing, which we will also have a whole chapter on that. So I'm not going to go into that too closely right now. And there are a ton of management strategies. So we can manage any issues that arise when the baby is in utero, inside of the mother. And there's different medications and supplements and therapies. So in summary, chapter 6. The fetal health involves this interplay between the fetus, the mother, and the placenta. Human development follows a highly structured pattern of development from conception all the way through fetal development. The placenta is a critical organ. It is nutritional, immunological, and endocrine functions that support the fetus. Deviations from normal development can result from the mother, the fetus, or placenta disruptions, and maternal care and screening is critical during pregnancies. So, it was a lot of information from chapters 1 and 2. Remember, it gets really granular. Don't stress yourself out about it. I promise I'm not going to ask you to detail in-depth mitosis. I promise. So let's go back to this week 1. So you've read the chapters. If you haven't already, you're going to. You'll watch these 2 videos, one on common chromosome disorders and one on myelination. You have probably already watched the Welcome to Our Course video. You're watching this video right now. Now you're going to complete your weekly reading quiz once you have read the chapters and watched this content. So it's 5 questions. Remember in the slideshow that you're looking for slides with the seal on it. It will be very helpful for you. And it's 5 questions. Sometimes it's multiple choice. Sometimes it's fill in the blank. Sometimes it's true and false. You have 2 attempts on that every week. Okay. So 2 attempts. Each question is worth 1 point, and your highest score will be taken. And then you will go to complete your online discussion. We'll touch base on that in a second. And then you will complete your survey on your field placement interest, okay? So your field placement interest survey, I'm just going to ask you a few questions about your location preference, your age preference, and then once I get that information, I can start working on where I'm going to place you for